Great. Thanks for having me. But rather recent work, but that's not so recent. So neural optimal transport for treatment outcome prediction. So it's the Nature method paper recently published. But I'm also going to speak, given that it's the biocontrol seminar, about fairly recent work where we move more to the control direction and diffusion models. So these are more technical presentations that still are basically targeted for biological problems or problems appearing in biology. So this will be the third part of the talk, just to give you a rough outline. What we see in the past years is that we really moved towards a personalized medicine. So the big question and the big task is that we want patients to receive really targeted therapies tailored to the molecular profile. And so the running example throughout the talk will always be somewhat cancer patients. This is due to the basically patient cohort study we are working with. And so this is going to be the running example, but these methods apply to also other diseases and other cases and other fields of research in biomedicine. But so let's say we have a patient and the patient has a particular tumor, and we are able to somewhat measure the molecular features of this tumor. So we have molecular properties of really single cells that give us a good insight in the tumor composition. Now we want to choose, right, for this particular patient, for this particular molecular profile, a drug that most effectively treats this tumor, meaning the tumor stops growing, the cells, aggressive cells are going to are getting killed, right? And we don't have a strong growth afterwards, or like forms of resistance appearing. And then if a new patient comes into the clinic, right, we have gathered knowledge about how different patients with different molecular profiles respond to a particular treatment. And then we want to choose for this new molecular profile the most effective drug for treatment. So that's the overall task. And what we've seen in the past year is really a revolution in our technologies to actually measure these molecular tumors, so these tumors on a molecular level. So we can, of course, understand the spatial architecture of these tumor tissues, but we can zoom in even further, understand the tumor microenvironment, or really understand of each individual cell, genomic feature, genomic features, and so on and so forth. So biology has now left us into a space where we can really say we have high throughput data, thousands of features for thousands of cells of an individual patient. And it's a unique opportunity to come in with methods that are machine learning driven to really answer questions such as how is a patient going to respond to a drug? What is the most effective drug to choose? And in particular, to provide a better understanding of the disease itself. What are particular biomarkers of a disease? What are good potential new targets for modern or for future therapeutics? So what we also see, though, is that the traditional machine learning algorithms not necessarily excel in this space. And so this is due to the very distinct nature of what we have here in personalized medicine. So one example, which has been a driving factor of the methods I'm developing, is that these omic technologies are destructive. What does it mean? So every time I measure a cell in order to get an understanding of the tumor composition of the tumor sample of a patient, every time I measure, I need to destroy the cell. So this classical setup right here, I want to understand how, given a particular treatment, a patient is going to respond, right? So I'm interested in the cell state or cells of the tumor before and cells of the tumor after in order to predict the effect. But this classical setup, or what I'm actually interested in, the response, is something I cannot measure because I need to destroy the cell when measuring. So I can only measure a cell either before or after, but I don't have per data of how a cell would look like after I give a treatment. So this classical dogma of machine learning, where we have paired data, like I have a input and I have an output, and I can basically just like regress or do supervised learning, right, on these paired input-output samples, is something I'm not provided with, right? I only have some cells which I choose to measure before treatment and other cells I choose to measure after a treatment. So we need to come up with machine learning models that are able to somewhat cope with these type of limitations, these special characteristics the data come with. And so what I did during my PhD is that I developed a framework that is able to do this. So on the one hand, we are able to reconstruct these dynamical processes cells undergo upon a perturbation without actually having access to single cell trajectories, right? So I only have snapshots of the population at a particular point in time, but each snapshot contains different cells. We also want to capture the distribution of particles, right? What we know in tumor biology, and we're going to talk about this later again, is that tumors are very heterogeneous. And we want to understand the heterogeneity because it's a driving factor on how a patient is going to respond to a treatment. So we cannot just look at average behavior over time. We really need to model the problem at the level of distributions over these different cell states. We want to develop neural network-based parameterizations because we want to be able to generalize to unseen cells. A new patient comes into the clinic, right? We want to be able to make predictions for this unseen patient. And lastly, we need to develop somewhat neural networks that are able to incorporate all these somewhat requirements as some form of inductive bias. So we need deep learning architectures that are really designed to modeling such population dynamics. And so there's one framework which somewhat is able to at least hit some of these nails or kind of fulfill some of these requirements. And this is something known as optimal transport. I'm going to talk in a second what this actually means. And so the methods we develop are basically neural optimal transport methods for modeling single cell dynamics. So what is also very beautiful, irrespective of its application, is that optimal transport has really nice theoretical connections. So it also is a very rich mathematical disciplines with, I think, 2 Fields medals, an Abel Prize, and Nobel Prize. So very nice theoretical connections to PDEs and dynamical system gradient flows. And also later on, and this is something we are going to explore throughout this talk, to control theory, diffusion model, Schrödinger bridges. So it not only comes with like very nice tools for us to utilize, but it also provides us with very nice connections so that we can actually explore very different, somewhat like different methods that are still basically mathematically connected. So there is an ICML tutorial from this year. Marco Couturi and I gave a tutorial where we summarize all these connections and their algorithmic properties. And also, if you look at my work, it some moves along the space. So the methods we develop basically utilize the connections between optimal transport, dynamical systems theory, and control theory. So in this talk, I want to basically tell you how we can predict treatment outcomes using neural optimal transport methods. I'm going to make this basically present you the Nature Methods paper from this year. Then we are going to basically skip part 2, where I usually talk about its connections to gradient flows and PDEs. But we are going to check or like understand how we can combine these OT methods with more control theory approaches. And in particular, why this is useful, because there's, of course, constant advances from the technological side that allow us to actually design even better models. And so this is something we are going to explore in part 3. So let's get started and let's dive right in. So just once more the setup. So we have a patient or a cohort of patients, and we are able to collect tumor samples of these patients. So now I'm able to do single cell measurements using single cell technologies such as image-based profiling or RNA sequencing. And I have now basically thousands of cells of a patient in a very high dimensional space or low dimensional space, depending on which method I use. And now I would like to be able to these cell states in this high dimensional space going to change or respond if I give a drug or any other type of perturbation. So I want to basically predict the change of these cells in this space. I only have access, right? I mentioned already these single cell methods are destructive. So measuring a cell requires destroying it. I literally need to open the cell to look inside. So I cannot measure the same cell before and after treatment. So mathematically speaking, I have a distribution that changes over time, a distribution over different cell states that changes over time upon perturbation, but I only have samples of a distribution mu before perturbation and nu after perturbation. I also mentioned that tumors are very heterogeneous, right? So they have very distinct cell states. Some cells might respond to a treatment, others might not. So just modeling the problem on an average level, right, an average population behavior, is not going to tell us what are the cells which are responsive, what are the cells which are acquiring some form of resistance. So we need to model really the problem on the level of distributions or on the particle, on the cell level. So somewhat, we don't have an alignment anymore, right? We have now samples of a distribution before and after. Somewhat, we need to reconstruct an alignment or understand which cell corresponds to which cell in order to learn an algorithm or like learn this map T that describes the perturbation effect, right? Before and like the cell states before and after a perturbation. And I can ask, okay, what is this optimal map, right? What is this optimal map that takes a point here on the left and maps it to the point in the perturbed space? What I know biologically is that all perturbations, of course, change some of the cellular features, but continuously in time, right? So if I measure close enough in space, the most likely map somewhat finds a matching so that the overall distance here, some form of cost between the cells, so the matching like with all the overall distances that is minimal might be the most plausible map to find. So given some cost function and finding basically the mapping, right, or a map corresponding to the minimum transport plan or the minimum alignment cost or misalignment cost is probably the one which is the most likely map to reconstruct this biological phenomenon. And so here I'm basically looking for the map T that minimizes this misalignment, so this mismatch here, given some choice of cost. And so this, in fact, corresponds to a very old principle in biology, in mathematics, known as optimal transport. And this theory actually goes back to a guy called Gaspard Monge, who lived at the same time as Napoleon, 1781. And so since then has been a lot of developments in various parts, mathematically and so on and so forth. And we are also not the first ones to basically use this minimum effort analogy or this optimal transport hypothesis to model cellular development. So there has been previous work at the Broad by Jeffrey Schiebinger or Caroline Uller, and they also made very similar hypotheses. So here the difference is that we want to learn this map T as a neural network. We want to parameterize it as a neural network. So during training, I have both samples of a patient before and after perturbation, and I can use this to now parameterize this map by a neural network, or here these neural optimal transport methods, because this is supposed to be an optimal transport map. And now I can learn this given these unpaired, right? Like I have from the same patients before and after, but I never actually have the pairing between cells. And then at test time, a new patient, right? I can use this trained network to predict how all these cells are going to respond upon perturbation. And here, this is basically my predicted outcome of the treatment. So let's talk briefly about this Monge map, about this optimal transport map, and how we can actually find parameterizations. So what we know theoretically is that this map somewhat exists under fairly general conditions. And also we are going to use one famous result by Bunne, which states that this map for a particular loss function, being the Euclidean loss function or the Euclidean cost function, that this can be expressed via a convex potential. So the map here is actually given by the gradient of a convex potential. So the equation I had on the slide before is in fact equivalent to this one, to the semi-dual, the argmin, now over the space of convex functions, and is basically the convex conjugate of the convex potential. And so now I can parameterize, instead of parameterizing here a map, I can instead now parameterize a function, and in particular here, I can parameterize with neural networks the space of convex functions. And so there's a class of neural networks constrained to this class, which is known as input convex neural network, which were developed by Brendan Amos in 2017. So this is just one way of actually parameterizing the Monge map. So this is something we did, I don't know, 2 years ago. The machine learning field moves fast. So there's by now plenty of other ways of actually parameterizing this neural OT method. This is just the one we are using in Cellot, but there's many alternative formulations. So here we have this convex conjugate. Just one level of approximation we make is that we just parameterize this instead of actually solving this. We just parameterize this by another convex neural network. So we have basically a pair now of two convex neural networks, which we parameterize. And that give us now a way of basically predicting, using this gradient of the convex function, which we parameterize by a neural network, how the cells are going to change upon perturbation. So this is the model we are going to learn. And so this is fairly similar for those who are familiar with generative models to other generative models, such as the Wasserstein-Gahn. So the Wasserstein-Gahn is doing this just for a different loss function. So this is the core method of Cellot. And let's look a bit into results. What does it give us? So the way we evaluate is that we have samples of we have cells in a control and in a treated state. And we have this for a screen of several 30 different cancer drugs. And now we can just use either image-based methods, such as the 4i microscopy, to actually look at the properties, molecular features of the cell. So here we have basically just we measure morphological properties as well as certain signaling activity inside the entire cell or just the nucleus. But we also have many of the results on single-cell RNA-seq data. So it works basically method independent or measurement independent. The collaborator we are working with and who is also involved in the kind of measurements for this patient cohort I'm going to talk about later, we just for now work with 4i data. So here we see, for example, for a particular perturbation for a particular drug, we see in the first two principal components, right, our cell population of unperturbed cells in light blue and perturbed cells in dark blue. And this is here just a biological system which we know very well to do some hypothesis testing, like does Cellot even construct what we know biologically? So it's a mixture of two melanoma cell lines, which are patient-derived, and we kind of know that some of them has developed resistances, the other one doesn't. So what we expect here and what we see is that some of the cells are not responsive to the drug, right? So they overlap heavily with the control state, while the others are actually showing quite a strong perturbation effect. So I can approximate this just here, just approximating the two measures, like the two distributions, and we clearly see here a strong perturbation effect, while this one is basically resistant to the perturbation. Nothing is changing. And so what we can do now is, right, we can just the method seeing the overall average perturbation effect. But then what we see clearly, right, here the map we would learn is the average, that also cell states which don't change at all would exhibit, we would predict a perturbation effect, right? And so this is something we don't want. We want to really capture heterogeneous responses. Similarly, there's previous autoencoder-based methods, and they basically expect, they model the perturbation effect as a linear shift in the learned latent space. But the way it's trained, it still more or less resembles population-based measurements. So again, right, we here predict perturbation effect for those cells which are actually not responsive. We don't make any, so this is our baselines, we don't really make any assumption on how the perturbation effect is supposed to look like. We are not saying it's a linear shift in a latent space. The only thing we say is find the map that corresponds to the principle. And then, of course, those cells which don't change at all are going to be mapped on top of themselves, while those who are exhibiting a strong perturbation effect are going to be basically mapped onto those cells. So we are able to capture or differentiate between cells which are perturbed or exhibit a strong perturbation effect and those which are not. So that's a very visually easy example, but of course we are also going to see setups which are less binary. Also, what we can look at is we can look at the changes, or if we capture changes on the molecular markers itself. So here, this is for two different markers, p-AKT, p-ERK. We can see the population of unperturbed states, right? So this is the overall distribution in the marker intensity. And then we see, okay, so there's quite a shift, right? Here is a bit less a shift in the overall population from unperturbed to perturbed. And what we want to capture is we starting at the light blue curve, right? We pass this to the network. We want to be able to perfectly fit the treated population. What we see is that the methods before, they really capture nicely the average, but just the average. And we want to really capture the population, like the entire distribution with its higher moments. And so this is our prediction. So we are really seeing that we are able to fit the population like like the heterogeneity of this perturbation effect. So what does it give us, right? Like, why is this useful? So now we can somehow subtract from a perturbed cell state the control cell state, because now we have a pairing. We have for each control cell, we can predict the perturbed state, and we can now subtract to really capture the perturbation effect. And we can do this for all 35 different drugs and really identify that all these drugs have very different and very distinct perturbation mechanisms. And so here, just zooming in, we see that for each type of cell, like for each different cell lines, right, which we can markers, we are able to see how different subpopulations are going to respond, for example, here after giving dasatinib, a cancer drug. So what are the feature changes? And it allows us to somewhat identify markers that are identifying, or this might be like that might be important to recognize or like to characterize a particular perturbation effect. So this is all not very, I mean, that's nice and great to see, but this is all not very interesting, right? So this is still cell lines. So now we move to predicting treatment outcomes on unseen patients. So we have a cohort of distinct patients for which we learn somewhat a generalized perturbation model. And then if a new patient comes into the clinic, we take this model to predict the treatment outcome. And so this is all part of a huge initiative in Switzerland known as TUPRO, Tumor Profiler. So the paper is not out yet. It's about to be out, but so I can already show some initial results. But so one core thing we realized by just like throwing Cellot on it is that these patient cohorts, of course, incredibly heterogeneous, right? So many different factors, such as genetic meta-information we have, like driver mutations, or certain other properties, such as the location of the tissue and so on and so forth. They are all going to somewhat influence the perturbation response or the response of a patient to a treatment, right? And instead of finding in my unperturbed population, now subpopulations of groups of patients for which we could predict or could learn separate transport models, right, given their properties. Ideally, we would just like to condition the learned transport map on this additional information we have, on the patient meta information we have. And then we can also use this current, we actually learned 34 different maps, OT models for each individual drug. But we could also use this to just learn one model over all drugs and just condition them on whatever drug we apply. So here, for example, trametinib, or we can condition on this is going to be dabrafenib, or potentially we can condition, oh, I'm giving a combination therapy. So just to show you that generally we could basically generalize this model to these type of scenarios. And so the way we do this is that we now parameterize, instead of like parameterizing a single map T between mu and nu, we are learning now context-aware Morse maps, or a family of context-aware Morse maps, that for each particular context, for each particular setup or set of meta-information, returns me the corresponding map that describes the evolution of a distribution mu I to a distribution nu I. So here I have a patient before a treatment. I give, for example, drug A. This is going to be with the response, but if I change the context, I change which drug I apply, then the overall response will also change. And so before we were parameterizing these Morse maps as gradient of convex functions. Now, of course, we need to have an additional input parameter C, right, the context. And we only need convexity or input convexity with respect to the cellular input, but not with respect to the context. So here we basically use a neural network that is partially input convex, just as some technical background. And so what we see now, moving from the cell lines to the melanoma cohort, so here we have 47 melanoma patients, is that we are still able, I mean, there is some decrease in performance, but we are still able to capture the treatment effects across different patients. And what is in particular exciting is, so here we just see like CELOT referenced against average and 2 other neural network-based methods. We see that for most of the treatments, right, every time things are blue or white, we are basically as good as other methods. We are really able to do this consistently well across many different drugs and for different metrics. Here we have a distributional metric. Here we are just looking at particular features. And so then we are also able to somewhat for 2 very different patients capture these perturbation effects. So here again, we want to move from light blue to dark blue. And so what we even see here is certain bimodal behavior, right? And we are able to really capture this bimodal behavior upon perturbation. So that's very exciting. And then we can start, like, given these type of knowledge, right? Again, now we have a mapping between a predicted cell state of a patient and its control state. We are able to do really nice analysis and have an idea of somehow association power of variables of interest. So what are particular here, what we have are these clinical features. What are particular clinical features that seem to be relevant? Because this is something which seems significant from the predictions we make. And this is something you cannot just see if you only look at the predicted cells, if you only look at the perturbed cell state. You really need to be able to subtract, to capture only the perturbation effect in order to see these somewhat correspondences. Similarly, here we are able to, we have survival rates for the patients. So how many patients are still alive after a year after treatment, right? We are able to understand, are there particular cellular states that are somewhat correlating like survival, right? Are this very aggressive subcellular states that are somewhat correlated to the survival of a patient. So these are very nice analysis we are able to make given these CELOT predictions, just to give you a few insights on why this is useful. So just moving to, so this is the results, and so this is CELOT, and so this is CELOT currently tested on this observational clinical cohort study. But so most of my work has been still on method development, and so far we've looked at a kind of easy setup, right? Like something I call static optimal transport. We have a distribution of unperturbed state, and we have a distribution of perturbed state. And so far we just looked at a single map T, parameterized it by a neural network, and just say, okay, predict me for each cell now the perturbed state. So it's really like a one-step procedure. But often, right, we are able to measure multiple times throughout the experiment. So we have an experiment at time point 0, right? And we are able to just sample some cells and measure them. And then later we extract a few more cells and we measure them, and then later again. So we actually have multiple snapshots across time. And also what we know is, right, all these processes are continuously in time. Like it's not just a one-step behavior. And ideally, we would really like to understand and predict individual trajectories that change over time. So that's the task now. So we need to somewhat reintroduce time into our analysis, right? So what we kind of removed before. And so there's multiple ways of doing this. And in particular, as I mentioned earlier, there are really nice connections of optimal transport to partial differential equations that allow us to really model these space-time processes, as well as stochastic differential equations. So really modeling these as continuously stochastic processes in time. And so there's a couple of works, but so for now, we are only going to talk about a work we've recently published in like UAI called SB-Align. So this also has another flavor, right? We are not only moving to this dynamic OT, right? We are also looking at a particular set of single cell experiments, which I'm just going to introduce in a second. So far, we've always assumed, right, or I told you that single cell measurements are destructive, right? I can only measure a cell. I need to destroy it. Every time I want to sample cellular process afterwards, I'm basically sampling, I'm taking a new cell, and I'm conducting a measurement. So this is, of course, like unique limitations of these single cell methods, and everybody is aware of this, right? So there's, of course, a lot of active research on how we can overcome this. So there's two developments. So there's a recent paper, and this is just one example of many developments in this space by ETH and EPFL and ETH, which is known as LifeSeq. The idea is now to only extract a bit of cytoplasm from a cell in order to conduct now from this extracted cytoplasm measurements, RNA-seq measurements, to measure basically the gene expression levels. But it's not going to destroy the cell because we are not, right? It's going to probably influence the cell, but it's not going to be lethal, right? And so then we can, from the same cell, again and again, extract basically this mRNA and conduct measurements. So now we have of a single cell, basically a continuous, or not continuous, but at least at distinct point in times, we have a measurement of the same cell. So we have somewhat an alignment, right? Similarly, we can also just use all these methods that tag the cells with genetic barcodes, which are going to be copied into the daughter cells. So like upon division, we actually have the clonally kind of identical cells that carry the same code. Example, at a later point in time, we find a cell with a particular barcode. We kind of know that they're clonally related, and we can still basically have an alignment in between. So here I call this partial alignments. Why do I call this partial alignments? Is these methods are either very low throughput. So here this means, in the setup of LifeSeq, this means literally poking a cell with a pipette. So that's a very manual work, and we can only do this for hundreds and not for thousands of cells. And at the same time, these barcode-based methods, they are somewhat very much affected by stochastic cell division, very much affected by sampling effects. So we also just we should of course use the alignment information we have, but we shouldn't take it for granted, right? Like it's given, but we want to have a method somewhat only like building up on it. But clearly we need methods that use this information in order to design the downstream algorithms. So now what we want, like the goal of this is now to construct or learn the stochastic process, this continuous time stochastic process, the cellular dynamics, using these technological advances, these partial alignments, or taking into account these partial alignments. And in particular, in order to do this dynamic OT, we are going to use a method known as Schrödinger bridge, which in fact is equivalent, or it's the dynamic formulation of an entropy regularized optimal transport. So we are still in the same set of hypotheses. And we are going to use this to learn like that describes the overall dynamics, cellular dynamics in time. And in particular, given these partial alignments, the contributions we make is that we are able to develop a robust algorithmic framework in solving this Schrödinger bridge. So finding this stochastic process P, given some starting position P0 and some like marginal at the end P1. And so it's very difficult to solve this. To partial alignments, we are deriving an algorithmic framework using decade-old ideas from math and computer science, so Dupes H transform, classical Schrödinger bridge theory, to derive a much more robust algorithm. I just want to use a few words about this Schrödinger bridge. So it's actually a concept which first popped up in physics, in particle physics. So Schrödinger, I mean, is the famous physicist of the wave equation. For 2 years in life, he was studying somewhat particle dynamics, thinking about what is the most likely random evolution between 2 point clouds of diffusive particles. And so what he came up with is that we want to find a stochastic process in our time, like kind of time window 0 to 1, such that the process at the very starting point corresponds to our marginal mu0, and at the end to our marginal, our measurement, right, mu1. And so we need to, of course, have some prior knowledge on the reference process. So we have some hypothesis on how these particles could evolve. I mean, this can be something very simple, such as just Brownian motion. But now we basically want to find the stochastic process that is most similar, or somewhat like, given these start and end constraints, somewhat similar to our reference process. Like it's the best explanation of these phenomena given some reference process. So this reference process has somewhat a bit similar role as initially when we had to choose a cost function. And so what is also known, and here I'm basically just, so the equation here at the end, so this minimizing this Kullback-Leibler divergence, we can also write it in a different way, which is just here as this kind of standard control formulation. We can basically just look at this as a dynamic. So this is in fact like a stochastic control formulation of regularized OT. So I'm skipping a bit the details, but we can really see this again here as our cost function, right? We over time here between 0 and 1, we want to minimize our cost, right? Given some control V, such that at the starting point, right, our stochastic process resembles marginal mu0, and at the end point resembles marginal mu1. And so here, at an optimal transport, we always have a mass conservation constraints, right? We need to conserve our mass. So I'm skipping a bit. But so basically, there's an equivalence of this dynamic OT. So we can write down this dynamic OT formulation as really a stochastic control problem. So here, if we basically have zero noise, right, we just have our particles at time point 0, at time point 1, red and blue, right? Optimal transport would just connect, right? Find the best alignment between them. So now this is time resolved. So here from 0 to 1, we have this like time gradient from blue to red. So this is now this dynamic OT, like over time, basically the particle moves along this line. And the more noise we add is just basically just making this as so this is now corresponding to a stochastic fusion process over time. So this is what we are trying to learn, right? So this is our cells at time point 0, our cells at time point 1. And it's going to give us a dynamic description of how our cells evolve in time. So now this is a very simple SDE class. We can just so this would just correspond to some Brownian motion with a bit of blur around, right? We, of course, would like to somewhat have a drift term that steers our dynamics, which we, of course, don't know. We want to learn this drift, right? And we want to just fix a diffusion. But so here, for example, a horizontal drift. So we can just look at here, just a random evolution of my particle cloud in time, just assuming that here the drift is just going from left to right. So, but now we need to basically parameterize. So the bottom line of before, right? So this Kullback-Leibler divergence system can be rewritten here in this. The task is really we can describe this system as a system of stochastic differential equation where we would like to learn the drift, and the drift is going to describe the dynamics of our cells over time. So now the stochastic process is given by this SDE. Task is to learn this drift. And what we also know is, right, given our data, we know that we have an alignment, right? And we know we can, for a certain x0, like a cell at time point 0, we know, right, that it's going to end up at this particular x1. So we can now reformulate this SDE into an SDE that is conditioned to start at x0 and end at x1. What we have then is an additional term of this Stoops H function. Similar, what we know from classical Schrödinger bridge theory for this reference process, which is also given by an SDE, we know that this reference process that is conditioned to start at x0 and end at x1 is given by an SDE where we have basically like a closed form solution, like a closed form expression of our drift. And now we simply need to regress the drift we want to learn to the drift of our reference process. So the reference process, which is describing them like kind of a valid evolution of our cells in time. So we know actually that these are equivalent for they have like a constraint, like an SDE constraint to start at x0 and end at x1. And so this loss function is now what we need to parameterize. So this is very simple. It's a regression-based objective. And it very much resembles all these score matching objectives that have been very successful in these diffusion-based or score-based generative modeling, if some of you are familiar with it. So this is a loss, or just like something very easy to train and very, very, very powerful with powerful architectures from the generative modeling space. So we use this now, we learn this drift expression, right? And this gives us now a model that describes the dynamics of our cell continuously in time. So what we can do now here, for example, for a fate determination process in hematopoiesis, we can look at our cells at day 2 and really continuously in time predict how these cells are going to change into distinct cell states at day 4. And what we also see is that we can look at day 2 we have a population of fairly undifferentiated cells. And then over the course of time, we really see all these distinct cell types popping up. And we are able to really capture some of this emerging heterogeneity in time. So we go from undifferentiated cells to various cell types. So that's something we have tested way less than CELOT. So CELOT was much more explored and tested on very, very different data sets with a certain biological kind of biological proofs by developing a cell type system where we kind of know the behavior. Here, this is just on existing data sets, and the focus of this work was much more on the method development end. But and this already concludes my talk. So this was the 2 methods I wanted to highlight. One example more from a control theory perspective, one example really from this neural OT perspective. And so big thanks to my PhD advisors at ETH, Andreas Krause at Apple, Marco Couturi, my host at the Broad back then. In particular, most of the work has been in very close collaboration with Lukas Pelkmans at UZH and Gabriele Gut, as well as Gunnar Rage and Stefan Stark. My PhD was funded by NCCR Catalysis. And from now on, any presentation you see me will be on projects I developed after my PhD. Thanks a lot.